Good morning, everyone. Welcome back to OmicsLogic Transcriptomics. Today, we will continue speaking about single-cell transcriptomics and also discuss some of the project ideas, if any one of you has had a chance to start thinking about those. So as we begin, let me just briefly show you a couple of things. First of all, I think everybody's making good progress. As you can see in the past several days and over the weekend, we've had some progress. So that's encouraging and that's really good to see. And I also wanted to mention that now on the forum, there is a project forum. So if you go to the forum right here, you will see on the main forum page, there is now the top topic, which is a project form. So you can look at that form to kind of get an idea of how to submit a project proposal. Now, the concept here is that throughout this program, we have been working on several different examples that we prepared for you to try out different types of data sets and different methods of analysis. And hopefully what became clear from that is that you have a number of different approaches that you can use to understand how mRNA is associated with a condition. So the first example was patient-derived xenograft models, and there the focus was really on how to map the reads to different genomes, to the human genome for the tumor, to the mouse genome for the mouse microenvironment. And by mapping those on two different genomes, we can start asking the question, well, how are they working together? What kind of defines different subtypes of cancer in the context of both tumor and the microenvironment? We then looked at the cell line data, which really showed how cell line from human genome, of course, cell lines do not represent the full complexity of system biology that we find in patients, but nevertheless, they are a good approximation to ask biological questions and train algorithms to start identifying specific features or specific genes that their patterns are associated with different subtypes of breast cancer. And today we'll speak a little bit more about single-cell RNA-seq, and single-cell RNA-seq will follow similar trajectories in terms of analysis. We'll see how the data is prepared and what are some of the specific analysis challenges associated with single-cell RNA-seq data. But nevertheless, it still helps us identify specific variation that is associated with a type. In this case, it could be a tissue type. And so looking at different types of tissues and different signatures in those tissues, we can ask more detailed questions, not just what is associated with a condition overall, but in the context of the complexity of tissues, which contain many different cell types. And then based on that difference, based on cell types, how do we characterize different conditions, right? So the level of complexity increases exponentially just by incorporating the differences between different cell lines. So on this forum, this project forum will help you think about a project of your own. So as we will go through today's session, I will briefly touch on some of these areas. If you go to this form, you will see here, starting from right about here, project title, problem statement, project summary, link for raw data, and reference publications are some key elements to get you started. It will help you do literature review and find data sets associated with different publications. So all of these would be publicly available data sets unless you have a data set of your own, in which case you can reach out to us about uploading that data and making that available for you. But the idea is to define the problem statement or whatever the project analysis you envision would be using methods that we have covered in this program and thinking about what might be the outputs and how will I interpret those outputs and what are the specific steps and settings I would have to do as I move towards that goal. So hopefully in the context of this program, this will be a step that will kind of solidify all the different aspects of RNA-seq that we have learned and will show you how in reality a lot of variation in the type of analysis will depend on what are the specific questions or the problem that you are studying. And also each step of the way, you will find what are some of the opportunities and the limitations as you go through this process of developing your independent project. Okay, so there's a question from David. How long are we going to have access to these workspaces platforms after the classes are over? This will help some of us who have had to do other things and weren't able to closely follow the sessions. So yes, you will have access for about a couple of more weeks. The specific date, I will find out from Beepsa. She's got the schedule. But in the next couple of weeks, you still have access to the asynchronous courses. You have access to the platform, computational platform. You have access to the coding exercises. And you also have access, so different people here have probably looked at different options that we had at the start of the program. We have kind of like a basic subscription and then a more advanced one. And that more advanced one will allow you for time with a mentor to help address any of the technical things that you might run into, and also to discuss any questions specific to your project. So in general, we are hoping that all of you will have the time to dedicate to your independent project, but we understand that it also requires time both on your part, and sometimes technical things can slow down your progress. So if you want to, and maybe you didn't even think about this at the beginning of the program, but you still would like to schedule a time to speak to a mentor, speak to one of us, help you address any of the technical issues and questions that you have, you can still do that. So we'll actually post on the forum also a request link where you can schedule your time. And then if you want to, we'll see at your subscription level and kind of figure out how to go from there. So again, any other questions that you might have, technical questions, conceptual questions, please post them on the forum. As you will see on the forum, many of you have already utilized the forum to kind of get responses, unfortunately over the weekend and things like that, right? We need a way to kind of monitor things and address them in a timely way, and this forum is a great way to do that. Okay, so so far, okay, any other questions before I continue? Okay, so far in Transcriptomics 1, we learned about mapping and quantification. We also looked at how to explore the table of expression. In Transcriptomics 2, we focused on statistical analysis. How do we compare conditions and know for sure that the patterns that we are selecting to interpret the biological differences are consistent between groups and within each individual group. In Transcriptomics 3, we started talking about mining that data to find more defined groups and variations within the full data set, and classification. So how do we take a machine learning model, train it, and use its prediction to understand what we can learn about other types of data based on a training data set that we have created. We also talked about feature selection, or how to select specific features, or genes in our case, or isoforms, that help define different types of classes or conditions or subgroups in our data set. Now, what's special about Transcriptomics 4 is that it really continues to speak about mRNA expression, but we have to kind of go back to where we started with Transcriptomics 1, because the way the data is prepared is different from what we have done up until now. So over the course of the past decade or so, there has been a tremendous growth in the number of different library preparation techniques that have been used to generate single cell data. And a lot of this had to do with lab automation. So the first manual approach was published in 2009, and the process was really time-consuming, manual, not very good quality data came out of that. But it was the first understanding that heterogeneity in the types of cells plays a big role in what kinds of questions we can ask and how well we understand the biology that we are measuring in bulk RNA sequencing. And so today we have a number of more established technologies. Some of them you're probably more familiar with others. 10x Genomics has kind of grown into its own field with some proprietary software and an automated machine that prepares the library, and then sequencing is done on the Illumina machine in any case. But you can see a review of these different technologies right here. It's an article called Exponential Scaling of Single Cell RNA-seq in the Last Decade. This is an archive PDF, so you can take a look over there. What is the process of preparing single cell RNA-seq? A big part of single cell RNA-seq is how the data is prepared. Basically, the question, how can you separate the cells for sequencing? The methods can be categorized in different ways, but the two most important aspects are the approaches to quantification and capture. So for quantification, there are two types, full length and tag-based. The former tries to achieve a uniform read coverage for each transcript, and the tag-based protocols only capture either the 5' or the 3' end of each RNA. The choice of quantification methods has important implications for what types of analysis the data can be used for. In theory, full-length protocols should provide an even coverage of transcripts, but as we shall see, there are often biases in the coverage. The main advantage of tag-based protocol is that they can be combined with unique molecular identifiers, or UMIs, which can help improve the quantification. On the other hand, being restricted to one end of the transcript may reduce the mappability, and it also makes it harder to distinguish between different isoforms. So again, you're limited to just measuring gene expression. The strategy used for capture determines throughput, how the cells can be selected, as well as what kind of additional information besides the sequencing that can be obtained. So the three most widely used options are microwell, microfluidic, and droplet-based methods. For well-based platforms, cells are isolated using, for example, a pipette or laser capture and placed in microfluidic wells. So you can see these wells right here. One advantage of well-based methods is that they can be combined with fluorescent activated cell sorting, or FACS, making it possible to select cells based on surface markers. This strategy is very useful for situations when one wants to isolate a specific subset of cells for sequencing. For example, you want to focus on just one tissue cell type, and then you can only take those from all of the tag-based selection here in the microwell. Another advantage is that you can take pictures of the cells, and the image provides an additional modality, and a particularly useful application is to identify wells containing damaged cells or doublets. So this information is useful in preparing the data for sequencing. Microfluidic single-cell RNA-seq, for example, FluidDigm C1, provides a more integrated system for capturing cells and for carrying out the reactions necessary for the library preparation. Thus, they provide a higher throughput than microwell-based platform. Typically, only around 10% of cells are captured in a microfluidic platform, and thus they are not appropriate if one is dealing with rare cell types or a very small amount of inputs. Moreover, the chip is relatively expensive, but since reactions can be carried out in a smaller volume, money can be saved on reagents. So you can see here that basically the idea is that you put your medium right here, and it flows through this path, and then the cells are separated. Here you can see how it works from a more of a diagram point of view. So the cells based on different type are located inside these little wells over here. So the idea behind droplet-based methods is to encapsulate each individual cell inside a nanoliter droplet together with a bead. The bead is loaded with the enzymes required to construct the library, and each bead contains a unique barcode which is attached to all of the reads originating from that cell. So all the droplets can be pooled, sequenced together, and the reads can then be assigned to the cell of origin based on the barcodes. So these droplet platforms have the highest throughput since the library preparation costs are around 5 cents per cell. And sequencing costs become the limiting factor. So a typical experiment requires some minimal coverage. And with this low coverage, only a few thousand different transcripts can actually be detected. So if you have an efficient droplet-based preparation of single-cell RNA-seq, you can put in a small amount of data without separating it, so there's not that manual process involved. It is tagged with barcodes, and then you can, depending on what coverage your sequencing is, determines how many individual features, genes, or transcripts are going to be captured in the final output. So as a result of this process, single-cell RNA-seq includes several steps of quality control and normalization. Because there's a small amount of material per cell and the sensitivity of sequencing, unreliable data is best excluded from analysis and the rest of the data made comparable using normalization. So broadly speaking, the analysis can be separated into these 3 different parts. One, dealing with the raw reads by checking quality and aligning to a reference genome. 2, dealing with cell-specific data by quality control, normalization, and elimination of artifacts. And then analyzing, or number 3, analyzing the resulting data set for differences using differential expression, clustering techniques, or network analysis, which typically is used for time course data. So again, if you imagine the specific challenges are that you have many different cells, you have low amount of input material, and also your variation, therefore, is going to be harder to achieve, right? So if you're looking at differential gene expression, as you remember from differential gene expression in bulk RNA-seq, it not only looks at the difference between groups, but it also looks at the consistency within each group. So here, as you have many individual cells and the way that they have been prepared has been somewhat different, that process of preparing the reads and then normalization of those reads is going to have an impact on what you can actually study in the end. So some of the important parts, we'll talk about kind of drop-seq from now on, is that the FASTQ files originally also contain the reads, and then for alignment, we have the cell barcode and what's called UMI. So UMIs are the adapters for each mRNA. And according to different UMIs, you can determine how many mRNAs are present in each cell instead of counting reads. So we count these UMIs in each cell. And this is called, kind of inferred to be an expression of that mRNA in a cell. And these unique molecular identifiers are short 4 to 10 base pair random barcodes added to transcripts during reverse transcription, so during PCR, RT-PCR. They enable sequencing reads to be assigned to individual transcript molecules and thus the removal of amplification noise and biases from single cell RNA-seq data. So that's called demultiplexing. So you have the raw data that comes in, and then you have these identifiers, and what you want to do is you want to understand how to extract the specific genes. Here you see these are genes, and how do you find them within the thousands of cells that you have. Then you can see that this specific step of counting UMIs is kind of the alternative of quantification of mRNAs. So you're looking at the barcodes and not the coverage as we did previously. So an important part of single-cell RNA-seq is cell quality control to actually understand that the cells were intact, and then you can also do that computationally. So as a result of that, you focus a lot on preparing data that is somewhat reliable, and then count the UMIs instead of genes, or expression of genes, level of expression of genes. And so what happens in the end is you have this normalized data that is ready for some kind of downstream analysis. So the processing is very different, but then you focus on this downstream analysis. And one of the features of the output table of expression is that it is called what's called zero inflated data. So zero inflated data means that in a lot of samples or a lot of cells, instead of knowing some level of expression, you have zeros. Now, if you remember back to when we talked about different methods of statistical analysis and machine learning analysis, we always talked about preparing the data for that analysis. And one of the important parts are intercorrelation within a data set, or also the properties of it being continuous and normally distributed. And so here you don't have the normal distribution. You don't have continuous form because you have a lot of zeros. And also, when you have zeros across many of your samples or many of your cells, they are just naturally intercorrelated. So you run into some specific challenges for differentiating between groups. But still, there are specific clustering methods that can help identify cells that are similar to each other, and then you can assign them to a distinct group. This is a lot of times using combinations of methods. For example, you can use PCA or t-SNE, but a lot of times they are combined together. So you first reduce dimensionality and then run t-SNE on that reduced dimensionality data set. Another possible analysis is what's called trajectory analysis, which is going to be possible if you have time series samples. And so in a lot of times you're trying to understand differentiation of cells. And so if you have multiple time points, you have sampled them, then you do sequencing, you want to understand how did the cell differentiate into different types, right? So you started from, let's say, some one common type of cell, and then eventually you end up with multiple types. And so then the question becomes, whether you have these time points. And this could, for example, measure response based on a specific cell type or differentiation, like I mentioned. So the analysis, kind of as an overview, is that you have your mapped reads. Those are mapped with barcodes for specific genes that produce these molecular counts. You check for artifacts, then normalize the gene coverage, and then you prepare a similarity matrix based on these molecular counts and develop a cell-to-cell distance matrix. And then based on that matrix, you can perform downstream analysis. So David is asking, how do you identify the same cell type over multiple times in the trajectory analysis? Well, as we mentioned, based on your data preparation technique, if you have multiple time points, one useful way to do that is using microwell with fact sorting. And so you are looking at, portion of cells that then differentiate. Or if you're using your bulk, right, you can also look at how the cells, you try to take a homogeneous cell culture, for example, right? So you take a cell line, and maybe these are stem cells, so they're all the same in the beginning. You know that just biologically. And then as you do single cell analysis, you look, how does that start turning into multiple groups, right? So you're not artificially tracking those cells. It's just the fact that you're starting with one cell type in the beginning, and then they start separating into more and more groups. So that's how you would, do that. And then network analysis is what allows you to kind of infer this ordering or trajectory, and it's a computational method of looking what is the likelihood based on the observation that you have at each time point for one to be related to the other. Does that answer your question, David? Okay. Okay, so again, a major part of this is understanding what are the challenges that are being solved and how can they be solved using different types of computational methods. So let's take a look really quickly at what types of data we can find for single-cell RNA-seq on NCBI. So please go with me to NCBI. Okay, and then here we can do single cell RNA-seq. Okay, and here, for example, you have 32,000 geo data sets, and this is processed. So we can just take a look here and also take a look over here at bioproject. Okay, so for example, you have here, we're just now looking at how was the data prepared, just to kind of understand what types of techniques were done to prepare the data for analysis. Okay, so a lot of different data sets here. Okay, actually what I wanted to look is at something relevant. Okay, so here we have single nucleus RNA-seq multi-omic profiling reveals age dynamic regulation of host genes associated with SARS-CoV-2 infection, or age-determined expression of priming protease TMPRESS2 and localization of SARS-CoV-2 in lung epithelium from a mouse. So a lot of interesting data sets, a single cell atlas of the peripheral immune response to severe COVID-19. Okay, so let's take a look just over here and let's see. Okay, so and again, what we want to find is the design. So how was the data designed? How was the experiment designed? So you can see here using 10x genomics to measure single cell RNA sequence to comprehensively characterize the lung microenvironment in 4 lung tissues from the same COVID-19 severe patient. So again, you have one organ, the lung, and then you have specific tissues from the same COVID-19 severe patient. And you can see that they have here the SRA experiments. So it's 4 SRA experiments, and then you have 1 geo data set. So if we click right here, okay, we'll see what this data looks like. Okay, and you can see over here the raw data. So what happens is that, the platform is going to stay the same. So you will still use sequencing using Illumina sequencer, for example, but you will prepare the data in a different way. And so you will end up having, an individual data set here. So if you look at how this data is organized, you can see, here's the full, TAR data set. And you can also look at SRA run selector to kind of understand what the data look like at the raw, SRA option. So if we look at the metadata, okay, so again, what is the approach that they took? 10x genomics would be droplet based, and you can see that they have they specify here total cells, so they did not select for any particular type of data, and took the full lung tissue and kind of looked at what all types of cells are going to be present. Okay, so does that kind of make sense? Does anybody have any questions about what these methods here mean and how to find these data sets that will give us some examples of what the data would look like? Okay, so if there are no more questions, I do want to remind you that in terms of having your input data, you will actually end up with 2 files for each sample. So in your, my question is on the indication of single cell RNA. So yes, Aditayo, please go on with your question, and I will in the meantime share with you the data that we'll use for our analysis. So what is the question, Aditayo? Okay, so let me know once you've got your question there. Does he mean the applications of, okay, so what are the applications? Yeah, the applications, as you can see, you know, for example, what is the purpose? Okay, so what is the purpose of, you know, this one that we were looking at? So you can see that COVID-19 may progress into acute respiratory distress syndrome, or ARDS, with high mortality risk. Its exact pathological mechanism, therapeutic obstacles, and the clinical sequelae, or sequences, I guess, are critical and unresolved issues. Here, we reported a representative COVID-19 induced ARDS case, experienced initially stable, then suddenly deteriorating up to final respiratory failure course until his death despite of lung transplantation. His lung pathology showed necrosis of parenchymal tissues, extensive immune cell infiltration, and lung fibrosis. Single-cell RNA sequencing revealed various immune cell populations that were largely expanded in his lung and manifested inflammatory activated functions. We also showed that cell crosstalk between lung macrophages and fibroblasts promoted pulmonary fibrosis through IL-1B and TGF-B signaling pathways. Although SARS-CoV-2 RNA remained undetectable in his respiratory tract specimens, including BALF at the later stage of his disease, the presence of SARS-CoV-2 was definitely confirmed in his lung tissue. Thus, this case indicates the pathological mechanism of severe COVID-19 that includes pulmonary SARS-CoV-2 persistence, deranged inflammation, and the extensive lung fibrosis, which set the barriers for effective treatments and indicate potential health complications for severe COVID-19 patients. So again, the application in this example is that when you have different outcomes for patients that seem to have a consistent type of infection, so right, for example, the virus ended up in the lungs, it goes into the lower lung, and then it causes this severe immune response. You would like to understand how that happens and what might be the barriers for treatment, right? Because, for example, inflammation can be treated. You can reduce the level of inflammation. But in the case of lung scarring, which is lung fibrosis in this example, the scarring, so these are dead cells that kind of prevent the treatment from getting to the cells that are being actively infected right now. And also, the severe immune response actually builds up in this mucus form that prevents oxygen to come through. So you have these two processes that you would like to understand. You know, how does the scarring happen? What tissues are affected by the immune response? And how does that impact the process of, you know, treatment getting to the cells that need that treatment most? Okay, so this is just an example. You know, again, what are some of the other applications of single cell RNA-seq? You can look through here, you know, many different projects, 32,000 different geo data sets that you can just kind of go through and understand what might be some interesting ways that people have been using single-cell RNA-seq. I mean, if you think about it, and you want to understand a population of people in the hospital, you can say, let me measure temperature for all people that are at the hospital. But does that really tell you, the difference between patients and doctors and nurses and people that clean the hospital and people that cook food in the hospital, right? You have all these different people that, serve the sick people, and then sick people could be of various different kinds. So if you were to just measure temperature across the whole hospital, it would probably be average, just the same as people outside the hospital. Maybe if you had a lot of patients that had high temperature, right, that temperature would slightly rise. But you would still lose the detail of the specific examples of the types of people that ended up in hospital because of high temperature. So now if you want to understand what types of individual patients do I have in the hospital, you might first want to separate them into groups, then study temperature within each group, and that will give you a much more sensitive approach to understand who is who in the hospital and how to exactly differentiate between them. Does that answer your question, Adetayo? Okay, great. So here you have, well, let's go now to the platform, and we'll try to run a single cell RNA-seq pipeline. Okay, so please let me know once you've got those files downloaded to your computer. Again, the files were file1 SVL and file2 SVL. You can see that both of them are pretty small. This is just one sample. Okay, so maybe please put in like yes or something like this, so I know that you have downloaded those files. And then please go over here to single cell. Okay, so Adetayo, again, I pasted the link to the input files. And I'll paste it here again. So you just download, unzip them, and then go right here to single cell transcriptome. Okay, you can see if you open up this file, okay, you can see where this data came from. So let's take a look at exactly where it came from. Okay, so here you can see that this is single cell from a mouse, and for RNA-seq. Single cell suspensions were generated using the ACT-seq protocol and processed through drop-seq to produce single cell cDNA libraries attached to microbeads. cDNAs were amplified by PCR and prepared for sequencing through tagmentation. Sequencing libraries were generated through tagmentation using Illumina Nextera XT library prep kit. So these are, again, let's go to the full project. Okay, you can see that right here we have a number of input data sets. And so they are going to be, we took some of them. So basically just a couple of, so one sample, this is just one sample that we have. And this one sample is single end. Okay, so that's something that you need to remember. Okay, so here, this is a mouse. So use mouse right here. And here we will use these individual SVLs. So 1 and 2. So let's just take, so this is 1. And okay, there we go. This is 2. Okay, sorry. So both of them I see are right here. Okay, so we have _1, _2. So just remember that, you follow this logic every time we have a project. And now we can start the pipeline. So again, we start from here. We already reviewed this kind of process. So we first of all filter based on low quality cell barcodes or UMIs. Here you have some setting for what those lengths would be. And if you kind of read through the data preparation, you'll kind of see where these numbers came from. And then you can do mapping. So again, we're mapping on the genome. In this case, we're mapping on the human genome. Oh, sorry. Let's go. Here, and we'll use star single cell. Then we have to, using our UMIs, right, we need to assign which reads came from which gene, essentially. And then I need to identify cells by their cell barcodes. So the barcodes, remember, were attached in the beginning. They're in the form of these beads. And then we quantify ZUMIs. This is essentially, instead of looking at coverage, we are using these UMIs to count how many reads fall onto a gene, assuming that that will represent the gene. We know that there are artifacts. Remember that there could be broken cells, and we can computationally kind of understand that from looking at how many UMIs are present in a given cell. And the imputation basically assigns those numbers to a different cell by barcode. Okay, and this is based on a clustering approach. And then what we can do is we can look at similarity between different cells. So for example, we have different types of barcodes, and we have we also have this ability to kind of understand computationally which ones are most close to each other. So there are several different methods. We're not going to, well, okay, we can actually try one of these. But then basically what we'll have is a table of expression based on clustering. So we'll have a cluster number assigned to each one of these, or we can visualize all of our data. And this is kind of a preliminary visualization. So for example, if you use PCA, you can then do more downstream analysis with single vector decomposition here. And you can use 2 or 3. So just click on save and then on end. Okay, so again, depending on what the project is and what you are trying to do with the project, how was the barcoding done, how were the cells prepared, you have to read into all of those different aspects of single cell RNA-seq data to determine what type of analysis you can do here and what would be the logic of downstream analysis after you perform, after you have a table of quantified UMIs, and then genes, and then types of cells based on barcodes, but also maybe based on barcodes, based on marker genes that can help you categorize the single cell population into different types of cells. So for example, different cells. Oh, so the SVL files are just text files. They're just called double SVL. So another way to do this, I gave you 2 SVLs there. After you select mus musculus right here, you can actually, without upload, just do URL bulk upload, and then use the second one. Here, input files, so just use the second one. It will have 2 samples already in there, but you don't need any software to open them. Just change the ending to .txt or say open with text editor, and it will open it for you. but you can also use just the second SVL using this bulk RNA, bulk file upload. Okay, so how many of you were able to prepare this pipeline, or do I need to do it again? Does anyone need me to repeat this pipeline? Okay, for those of you that want to or will in the future look at different types of data, a common step when you get your output files, they come in this BCL format. So if you are generating your own data, for example, you're looking at 10x genomics, you might have this BCL file as your first output from the sequencer. And so then you can request this BCL2 FASTQ software. Essentially, it is a part of the 10x genomics suite of data where you can explore different types of gene expression. You can see they provide different types of analysis. So depending on how your data is structured, you might want to explore these automated ways of dealing with your data. So again, if you come across a data set, you spend some time looking at how the data was prepared, and then based on how the data was prepared, you can determine what would be the steps of your analysis and what would be the potential questions you can ask from that data. And I recommend just looking through some of the publicly available data sets first before you start going through a specific data set, just to kind of understand what's out there. Okay, so if everyone had a chance to start this pipeline, I just want to kind of give you a sense of what specifically in this project was being done so that we understand the context. So this data set is about the brain cells, neuronal circuits, and the cellular mechanisms and pathways that are involved in behavioral response are key for our understanding of anxiety-related behaviors in rodents. What you see here is this is taken from a brain, and the brain has these neuronal circuits that underlie fear and anxiety-related behaviors. So we can review some of these techniques in the context of how different cells that are involved in these circuits could be studied using single-cell RNA sequencing. So this is a microfluidic-based single-cell RNA-seq analysis. It's a newly developed method called ACT-seq that combines single-cell RNA-seq with a new single-cell preparation method that eliminates transcriptional perturbations that are artificially induced by conventional cell preparation procedures. So what you can see are these networks, which are essentially communication between different cell types. So you can see here, different examples or subsections of these relationships between anxiety, behavioral, and activity in specific neural circuits. So this is a view of the rodent brain that includes distal circuits implicated in anxiety-related behaviors. B is septohippocampal microcircuitry linked to anxiety. C and D are extended amygdala microcircuits involved in anxiety-related behaviors, including BLA projections to BNST and BLA projections to CEA. So essentially what we're talking about are the different colors are different types of cells, and they communicate with each other. So through this type of analysis, we want to separate the individual types of cells, capture their physiology, and then develop a molecular signature to understand how does this actually happen at the molecular level. So we can use different methods to study cell morphology, electrophysiology, and molecular signatures, and hopefully combine them all together to understand what is really going on. So first of all, these are kind of the outputs that we are expecting. And the relationship that we're looking for is the morphological and the functional properties of neurons. The sensitivity and how they function together is what we're trying to understand. And in this specific project, there's a combination of the fluorescence in situ hybridization, or FISH, so that we can look at co-localization of specific types of expression. So now, again, we are going through this process of RNA-seq processing. We build the pipeline. First of all, as you saw, we use, there are a number of different types of approaches. So we use star single cell for mapping on the reference genome. This is a mouse genome. And then we will look to assign those reads to UMIs, identify cells, then quantify UMI expression. This is like quantification of gene expression, correct for artifacts. And then in one case, we can do cell-to-cell similarity, or we can just visualize our results. But this is really just the beginning of this process. In the end, once we have our table of expression, we need to use marker genes to understand how to correlate the marker genes back to this type of a analysis, where we want to understand relationship between different types of cells and how expression of molecular genes associated with the signaling pathways in the neurons happens during a particular type of behavior, which is anxiety related. Okay, so any questions about this process? What kind of marker genes do I mean? Housekeeping. No, housekeeping genes are primarily used in microarray. So you know that certain genes are found in abundance. They are fairly stable in terms of their expression. For example, they are responsible for some basic function of any cell. And just to understand that you're properly capturing the signal, you use these housekeeping genes. Some of them typically have zero expression, some have medium expression, some have maximum expression, and that gives you kind of a range for microarray-based experiments. In single-cell RNA-seq, we are looking at different tissues. So different tissues, how do we kind of categorize tissues? We categorize them by cell morphology and function. And so if you have that phenotype, it is determined by specific genes that are more activated or less activated in a given cell type. So you can actually, you've probably heard of the cell atlas. So if you look for cell atlas marker genes, there's this whole project to develop the markers for individual cell types based on sequencing many examples, and essentially help, once we have changes in expression, right? The changes in expression, most of the time, when you look at differences between any kind of data, the majority of the differences are going to be between types of tissues or types of functions that these cells perform. And that's defined by these marker genes. Once you kind of establish what type of a cell it is, within that type, you will have some variation. And then between different types, you will have some variation, maybe not related to these specific marker genes, which gives you some sense of how they function specifically. Okay, and so David is asking, does that answer your question, Katerina? Yeah, so like, for example, we have samples from model organisms. They will have these marker genes, so mouse, human, Drosophila, etc. If you have a non-model organism, then obviously it becomes a little bit difficult. you still probably, expect that some related model organism would have those marker genes. Okay, so Katarina, let me know if that answers your question. And David is asking, is it not possible to do cell differentiation trajectories on a single time sample based on the fact that there will be a heterogeneous population with cells at different stages of differentiation? Yes. So again, when you say that there are different stages of differentiation, but you just take one sample at one time point or multiple samples at one time point, you don't know which one necessarily, differentiated or they were already like that. So that's why people do this kind of a trajectory based on. So for example, if you had, one sample had all, the same type of cells, then in the next time point you have 20%, and then in the next one you have 50%, then you know that the additional 30% came from the 20% somehow, and you can measure, how did that happen based on gene expression. Now, if you have just one time point, you just take a snapshot and you say, okay, there's 20 like this, 30 like this, 50 like this. And of course, you can make comparisons between them, right? But the differentiation trajectory implies how it changes over time. So I don't think you can do that from just a single sample. You can just study differences between differentiation stages. Yes, you can definitely study differences, but not a trajectory implies multiple steps along time axis. Okay, so what this really brings us to is this whole concept of developing an independent project, right? So now as we think about how all of this type of data helps us address different types of challenges, computational challenges, we need to think of how to develop a project. We need to think of how do we find a data set and maybe a reference publication and define exactly what might my project be so that I can determine what is the most appropriate analysis approach for the data that I have selected, for the environmental or experimental conditions that I'm studying, and how do I go from exploratory data analysis to kind of contrast that I identify, and then I have this comparison established and I can find some meaningful signal. So let's talk about choosing a topic for your project. I mean, I understand that a lot of you have already gone through this in maybe your other research experience, but let's just review this for the sake of doing it together as a group. So the best way to choose a topic for your project is to take topics that you know and care about. And you can kind of think, can I talk about this topic to people for more than 10 minutes at a time repeatedly? And that tells you that you know and you care about this topic, right? You don't run out of new information to talk about it. You're interested in new information for that topic, and you can talk to other people about it. And so you can also think about it on whether you can allocate significant amount of time in the next few months thinking about that particular project and dedicating time to actually analyze the data. As you probably have found, this is time consuming and sometimes tedious, so that motivation is very important. So the second part to think about is what will be interesting to your audience. So how will other people react to this? And here you might have an existing audience within your niche area that you're very focused on. Maybe it's your lab members, or you might want to think about a more general audience. And I think in both cases, you have to think in very simple terms to maybe even try to explain it to other people and see what kind of feedback you're getting. And then you also have to find a topic that already has been sufficiently studied and has some available data, because as you are starting, you want to get far enough into the project without having to generate new data right away and kind of understanding what others have covered in this particular domain. So you have some framework to think about the problems that you're going to be facing. So in the intersection of these three things is really where your project is. So your project has to kind of have these three elements that, this is a topic that you know and care about. it will be interesting to some audience, and you can define that audience. And then you also have access to data and some knowledge about this domain as you are getting started. That will help you get far enough along to be creative with what new information you might want to generate. Now, the second part or stage in this process is to find the right sources. And there are three types of kind of data sources. There are primary sources, which is when somebody generated raw and original data from their first-hand observation. Secondary, which means that there has been analysis, and this is a, maybe somebody put it out, already published an article, and they are, describing how they analyze that data. But that data, that raw data, you have access to it. And then tertiary is really a interpretation, so to say, of that data, where people have compiled multiple studies together. They kind of analyzed them in context, and now they are making those compilations available. So when people publish, they go from raw original data and all the way to potentially interpretation in the context of existing databases. What we are doing is really going the other way around. In our ability to take other people's data, we need to start from the context. So what's available, what types of databases, if it's cancer, let's look at the Cancer Genome Atlas. If it is COVID-19, let's look at NCBI virus, right? So let's look at compilations that have been already made available, then go in deeper and deeper until we gain access to this firsthand observation, and maybe then we will extract some meaningful insights from there and define our problem or question, right? So we start from a high level and then go lower to find a problem or a question. Because when you think about a project, the project is driven by finding a solution or an answer to a specific research question. So you go from a general background. So maybe as you are reviewing different sources, you might want to categorize them into different types of opinions and different types of studies. As we saw in this last example, this was an animal study focused on anxiety using multiple assays. So getting that perspective about anxiety and what those assays, what type of data they produce, you might start getting more into, molecular markers of some particular behavior and think about these neural circuits. Many times you will see that there are gaps in current research, unanswered questions, controversy, or debate around a particular issue. People will identify specific topics in these review articles that have been controversial or unexplored sufficiently to come to conclusive results. Then you will use that opportunity to define your own question or hypothesis, right? So that will really make your hypothesis specific, interesting, and a valuable addition to existing research. I think that one of the goals for us is to help go through that process. And what we have done is we have reviewed multiple articles to make sure that we can guide students and participants of our programs to go from selecting research articles to finding the data, understanding the methods, and understanding what questions are being asked. So some of these examples are going to be useful or maybe even sufficient for those of you that want to go beyond just kind of like the practical tutorials to really look at how this data could be informative. So to briefly speak about one of these projects that we recently completed, I want to turn your attention to the project tab. So if you look over here, right, these are the courses that we have, and then you can go right here to projects. And under projects, you'll find several examples. Some of them were briefly touched on in the context of the training sessions that we had. Some of them might be, completely different from what we already did. So you can take a look here at some of these educational projects. And, they are categorized, or, you could categorize them by type of data or even type of application. So for example, we have here a project on crop drought resistance. We have here, different neuroscience projects and cancer-related projects and things like that. But one of the projects that we recently put out there, and this is still something that we are revising. So if anybody is really interested in this, it's an avenue to explore the what's called TCGA, or the Cancer Genome Atlas. This is a project that was developed based on the TCGA data repository that describes clinical samples from patients and also from multiple types of omics data. But a lot of that data has to do with transcriptomic or RNA-seq data. So a lot of it is based on what we did in this program. So maybe I'll just post this link. Okay, great question. How to download data from TCGA. So again, this is a good place to start because it will explain how to download the data, how to find clinical information about the data, how to develop a sense of what clinical categories to look for, and how to then do the analysis with the idea of what type of clinical value this idea may be. So if you want to work with TCGA data, this is a great place to start. I also want to mention that, Harpreet, who has been mentoring you in this program, is the one that she, first of all, has a lot of experience with TCGA data, and she has been working on TCGA data for peer-reviewed publications herself, and she shared a little bit about that in her previous presentations. But in this project, she also broke it down into very specific comparisons that illustrate both some technical aspects of analysis and conceptual ways to think about how you might get to a meaningful analysis result. So briefly, I'll tell you about some of these, and then if you're interested, you can go ahead and try one of these projects. So a good place to start if you are thinking about TCGA, and by the way, let me first ask how many of you, and maybe you can just put what is it that you're interested in. So, if you're interested in a particular type of cancer, you can write it down, lung cancer or breast cancer or liver cancer or whatever. If you're interested in, COVID-19, so just let's put together in the chat box, what are you interested in, so we kind of get a sense from where we are starting. Okay, so ovarian and breast cancer, multiple myeloma, acute myeloid leukemia, vector-borne diseases, okay, breast cancer. Okay, anyone else? And I see, yes, someone here has a name in Arabic, so I can't read it. But if you could say what your name is, that would be useful also. Okay, Anita, malaria and COVID. Okay, so what about Marek, Krupa, Katarina, Jefferson? Okay, so at least a few of you are interested in cancer, and so TCGA might be an interesting data set for you to consider, and I'll ask for the rest of you to try and answer that as well. So, if you just look for TCGA, you will see the description of TCGA right here that explains what exactly they did and how this data was used. And here you will have access to TCGA data. And unfortunately, this portal, I personally do not like as much as some other portals that have essentially a replica of the same data set. So here you have to go through many different steps to actually find your data, right? So for example, here you can go to repository and you can start searching through, all of these individual cases. In other repositories, one of them is called Firebrowse. You can find this data much easier. So Firebrowse, you can see, is a broad project that compiles these different data sets based on different types of data and based on different types of cohorts of patients or different projects based on basically what kind of cancer they have. So if you're interested, somebody mentioned breast cancer, so you have this project called BRCA. It's breast invasive carcinoma. And you will see here different types of data sets that are available. So here you can find and download this data, including the clinical data, right? And that's really what's important in these data sets, is that you have, tumor samples. A lot of times you have blood samples or healthy tissue samples to give you a baseline, but you also have some clinical history about these patients. And importantly, I think that in these clinical terms, a lot of data is there, but also a lot of it is missing. And a more neat representation of this data typically comes in the publications that came out associated with the specific cohorts or kind of projects focused on different types of data. So, for example, this project on liver cancer that was made available here just recently is using some of the insights and methodology that has been published right here, comprehensive and integrative genomic characterization of hepatocellular carcinoma. You can see that it was published in Cell, so you can go to the original article here as well. They describe all of the strategies that they pursued and the questions that they pursued, as well as some of the outcomes from this project, from collecting all of this data, which essentially resulted in reclassification for some of the subtypes, including the different types of cancers driven by hepatitis and driven by NASH or genetic factors, as well as lifestyle factors like alcohol consumption, etc. So you can read in a comprehensive way, kind of a review and a comprehensive analysis, but also get access to data sets that describe in a more neat way the clinical data about the patients. Okay, for some of the other projects that you're mentioning, I recommend starting with NCBI. NCBI really is a great collection. I mentioned vector-borne diseases, cancer stem cells, different types of diseases like malaria and COVID. So you can really find a lot of this data right here. And there are two places that you should look when you search, right? So if we search for malaria, you want to focus in the context of gene expression. Start with exploratory data analysis first. It's always more useful to explore your data before developing a hypothesis. And you can explore data that is already prepared in Gene Expression Omnibus. So you can go to Geo Datasets. You can also look at BioProject. And BioProject is a collection of projects that has information about how the data was prepared and also what were all of the data sets that are produced, right? So for example, SRA experiments are those direct FASTQ sequencing files. And sometimes they will also have the processed version as a geodataset. Okay, so a little bit about how to think about your projects. And in our next session, we are going to review some of your ideas, further deciding how to move forward with the analysis. So again, the objective between now and our next session, which is going to be the last session, is to fill out this project form. Even if you are not planning to look for, if you're not planning to do your analysis or complete your analysis in the near term, just looking through the data sets and thinking through the analysis steps is going to be very helpful for you to kind of review all the different methods that we covered in the program, all the different types of considerations about how to set up those methods, and also think about expected results. How am I going to apply the method to find a particular result that I'm interested in? Okay, so any questions, again, put them on the forum. If you have a specific type of project idea, you can start filling out this form. This form is dynamic. In other words, you can fill it out with any gibberish you want and then change it. And we will get the latest notification about the change to help kind of talk through some of those things. And I will briefly, for the next 10 minutes, pass it on to Harpreet to speak about and a little bit explain better what are some of the ideas and data sets included in this TCGA project. Harpreet.
Thank you, Ilya. Let me share my screen. So, hello everyone. So, let me just explain exactly what this project is about so that even if you want to, when you are going through, so you must be familiar with this. So, first we have obtained a TCGA data, as Ilya has already shown you that have taken the data set from 5 rods. So, then our first, sorry, we aim various kind of objectives that we want to address. The first, when it is liver cancer, so whether we can differentiate it. Okay, so whether we can differentiate liver cancer samples from the other cancer types. It could be the breast cancer. So apart from that, if you are comparing, that is between the tissue. So then if there are non-tumor samples. So for that first objective that here we have done, that is pan cancer analysis. So here we are trying to compare uh, liver cancer samples that are tumorous and non-tumorous. Then we are comparing with the breast cancer tumor and non-tumorous sample. So for that, there are what could be the already, because you have learned a lot of things during this transcriptomics course. So what would be the methodology that we can explore? So here first we have applied principal component analysis. So to if I go to the principal component analysis, so this is like, if I'm just here I'm just representing you the results. So here we have mainly 4 types of sample. One is LIHC-TP. LIHC means stands for liver hepatocellular carcinoma. So as you might be familiar with the liver cancer, can be cholangiocarcinoma, it could be hepatocellular carcinoma. Hepatocellular carcinoma is primary liver cancer type. So here we are trying to first classify liver cancer primary tumor and non-tumorous samples. Then we have breast cancer samples and breast cancer non-tumorous samples. So here we just have taken all the data and then we try to whether we can differentiate based on the signature, whether we can differentiate those genes or not. So then once we have run the PCA, then we want to identify which specific genes are associated with a liver cancer tumor type or the breast cancer type. So if you are when we are going through this transcriptomics course, so then we come up when we have multiple factors to consider. So one of the best method approach could be that we can apply factor regression analysis. So then we have tried to apply factor regression analysis to identify which are important genes that are associated with liver cancer type or breast cancer type. So this is the second part. So here you will, we have first explained exactly how we perform this analysis. Then we have tried to identify which are genes that are actually significantly associated with the breast cancer, liver tumor, and the breast tumor. So, and what are the genes that are actually significant only the tumor between the non-tumor samples. So those kind of analysis we have done and then identify the signature genes. So once you have those genes that can differentiate these cancer subtypes, so we can say that we had instead of even the breast cancer, if somebody is interested in some other cancer type, so that that person can take that particular samples. So the I think the one of the advantage of specifically factor regression approach, that you can distinguish multiple conditions and identify the and consider multiple factors. Then after, once you have able to identify the signatures gene that are actually associated with the liver cancer types, so then we next proceed to perform exploratory data analysis. So the exploratory data analysis, that here we want to identify or the distinguish different clinical subtypes. So the clinical subtype, when we are saying that, that's like it could be the cancer can be classified into the different tumors, like so early stage tumor or the late stage tumor. So it could be like stage 1 or stage 2 or the stage 3. So for that, first we proceed for stage classification based on the gene expression data. So here, what are the approaches that we can explore? So how we can classify different types. So during this, machine learning is one of the approach which that we can use. So here we have explored the different for two classes. For the stage classification, we have explored different supervised machine learning techniques like random forest, LDA, naive Bayes, SVM. So this is how we first we have just shown how first of all we have identified important significant gene. So if you remember that during the supervised machine learning, one of the method that can actually select the features, that is random forest. So first we have run uh, this random forest on the all the genes. So after then, once we have identified important significant genes and how we can select those genes, then we have further proceed for, then we selected this subset of genes and developed various supervised machine learning prediction model and try to understand exactly which particular prediction model can classify these sample based on the stage. So then this is for the stage classification. Then the another factor that is important, that is there are various risk factors. So for the liver cancer, risk factor can be any hepatitis infections like hepatitis B or the hepatitis C. Then apart from that, it could be alcoholic condition. So those are like considered as the risk factors for the liver cancer. So the next we proceed whether we can classify the samples based on the risk factors. So here again we have applied supervised machine learning approaches and try to identify a particular subset of our genes that can actually distinguish sample based on the risk factors. So once we understand these two approaches, and then further we proceed for the multi-omics integration. So here we have tried to integrate two types of data, that is RNA and the miRNA. So here we have different approaches. Exactly our what was our goal, that all the machine learning methods or supervised machine learning methods, they are actually, when we know that this particular risk factor, we are actually considering a particular condition. So if you don't know and you want to explore all the data and try to understand exactly based on the multi-omics data how they are associated with the different clinical subtypes. So it's not we are like already filtering or some particular gene. So we're integrating the all the RNA and miRNA data and then try to understand exactly based on multi-omics integration, this data is actually representing the pattern of which clinical factor. It could be maybe the risk factor, it may be the stage classification, it could be a simple cancer that is gender based. So all those, whether multi-omics integration help us to understand the association of this data with any clinical factor. So for that we have explored this third part, that is third type of analysis where we have that is multi-omics integration. Here you will find one method we will need to update. So the one is that is uh, SNF method. So that is similarly that is neural network based method. So the next is O2PLS method. So how we can integrate multi-omics data, and you will learn all those approaches and exactly how we can drive biological meaning from multi-omics data. You will understand all this concept under the multi-omics data integration. So this is the actually the I am just giving you the overall overview of this paper. So beside we have also provided the exact all the links links for the data set that we are using for particular analysis, and we are also explaining it step by step in a step by manner exactly how we are performing any analysis. So even if you have unfamiliar or you have yet not explored any of the steps, so you will understand all those kind of analysis, and you will also understand what kind of analysis we can perform and what kind of methods are available to execute any analysis or to drive a biological significance from the data. So this project is all about this. So the on the starting part you will get the project overview and of course the TCGA project, and then what is hepatocellular carcinoma and what are the objective and all these methods. So you will learn all those things, and after that you have all the three or the four different kind of analysis. Even if you have your own data, after going through this project you will understand exactly how and what kind of analysis that you can perform once you have any transcriptomics or any multi-omics data. So if you have any question. So I think it would be better you will explore this project yourself. And once you have, I just hope that you have completed all the transcriptomics courses. Especially if you have completed from transcriptomics 1 to transcriptomics 3, you will find every single analysis, how you can perform any analysis. So the transcriptomics 2 and 3, 2 is specifically based on the statistical analysis, and the 3 is mainly about machine learning. So if you have gone through all those transcriptomics courses, and you will understand exactly what kind of analysis we should perform, on what kind of the methods are available if you want to perform any analysis on your own data. So beside, I would like to show you how you can download a data set from a geo. So let me just take you a platform. So if you are on this platform, so for example, you are interested in liver cancer. You can, depending upon your research interest, you can make that query. So once you created this query, here you will find all the this number, and just click on this number. So from here, first you just on the right side here we have this this organism. So you can select a particular organism that you are interested. So once you're able to select this organism, so then you can proceed under this study type. So from here, if you go to the customize, so you can exactly select a particular study design. So here we have, like, we are interested for high throughput sequencing data. So I'm selecting this particular option, that is expression profiling by high throughput sequencing. So I'm unchecked all other options. So let me just show. Once we show that, and then you just need to click on this. From here at the top, you will find this option, sort by number of the sample. So you can select whether you are interested in that is high to low or low to high. So most probably we always choose high to low. So then For example, here, if you look at, this is the single cell transcriptional data. So I'm just going to the another data, which is the second data set, which showing the deregulation of ribosomal protein expression and translation promote breast cancer metastasis. So here they must have compared both liver cancer and the breast cancer types. So here we have 344 samples. So if you click on this, so we'll direct it to this page. So here you will find that this particular data have multiple series, right? So like here we have, for example, we are interested in this series. So once you have a series data, so then you will find the detail exactly what was the data set that have submitted, exactly what was the analysis that previously is done on that particular analysis. So you can go through here, and under the summary you will find all the details. Under the overall design, you usually get information how many samples are there. Then here under the supplementary file, so like here in this particular data, they have already provided a read count value. So one way is you just go with this data and just try to perform your analysis, because already you have read count data. So, and then we have this series matrix file. In the series matrix file, they have, usually they provide clinical data as well as the gene expression data, whatever the analysis kind of values that they have. And from either you can go for this complete read count data, and otherwise this data have all the clinical information as well. So you can take this file. Then they have also provided the link to the bioproject and SRA. So if you are interested, like you want to generate your own pre-processing value, that it pre-processed value, it could be like if you are interested in the FPKM value or whether the read count value, so you can directly go to this SRA, and here is link is provided. So from here you can, if you just this directed to the SRA page, and from here you can send this FASTQ file, links of the FASTQ files into a file that make a like SRA run table. So you can generate that and create this file. It will produce your SRA run table. So this is how you can actually download any data set. So from the geo, you can directly download this matrix file or the supplementary data. And from the SRA run table, you can directly download this SRA run table. And then this run with this run table, we can perform RNA-seq analysis that we have when we run that on T-Bioinfo platform. Then you can actually pre-process this data, whether you are interested in to generate FPKM value or you are interested to run read count values by your own way. So this is how you can download data from these two public repositories. Ilya has already shown you how you can obtain data from the Firebrowse. So is it clear to everyone? In the next session, we will try to take an example. Okay, Francisco, that's great. So if you have any questions, or I think already Matthew have a question, we'll dot related the course posted in the forum or answer after the last class. Yes, we will answer your questions as well. If you have any query, so we will address your questions that you have posted in the forum. Still, even if you face any problem during any analysis that you are trying to when you are going through any courses, so we will try to address all those questions. Okay, so thank you everyone. So if anyone have any other question? So if there is no question, so now I can So we can end this meeting if there is no question. So thank you everyone for joining today's session, and we just hope that you will complete your just submit your project form, and we will go through your project in the next session, as well as uh, we will try to demonstrate to you how you can proceed any analysis once to directly obtaining from that you have obtained from public repository like SRA or the GEO. So thank you everyone.